Good morning. Today we'll discuss about chip assay. The first question comes to our mind, what is chip? In chip assay, CH refers to chromatin, IP refers to immunoprecipitation. The word chromatin. What is chromatin? Chromatin is nothing but DNA and protons. DNA plus histones are other protons. If you observe the black colored DNA and green colored histone is positive and DNA is negative. To understand binding between histone and DNA, please watch nucleosome video. And here the second word, that is immunoprecipitation. Precipitates, settles. Immunoprecipitation. Here you are observing yellow colored antibody, a red colored protein and an insoluble bead. So what is chip assay basically? It deals with DNA protein interaction. So protein of interest, if you want to find out which sequence has compatibility with protein, which sequence of DNA binds with protein, we can do this chip assay. Nothing but DNA protein interaction. Now we'll learn the methodology of chip assay. We are dealing with DNA, where DNA is present inside the nucleus of a cell. See, the first step is we need to add proteins to DNA. I mean proteins of interest. So how to do this? The first step is cross-linking. You can use formaldehyde or UV light to cross-link protein and DNA. So proteins and DNA are fixed now. The next step is you need to shear the DNA, sonication procedure or enzymes you can use. So later our concept, that is immunoprecipitation. What we are doing now in the third step, we are adding antibodies with insoluble beads. You can use magnetic beads also. Once you add antibodies, which reacts with the protein of interest, so the FAB fragment of antibody reacts with protein. See, in up and off tube, you are observing yellow colored antibody interacted with the protein. It settles at the bottom. So this results in immunoprecipitation. Antibody protein DNA complex. So later you can take out the sequence. You can do PCR, amplify the DNA, and with the help of sequencing, we can determine protein binds to which sequence of DNA. We can easily trace out protein DNA interaction. So here in the cross-linking step, you are observing protein interacts with DNA. So red color protein interacted with green color DNA. This is the first step, cross-linking step. Later, you are adding antibody. You are observing yellow colored antibody with an insoluble blue color bead. You can also use magnetic beads. Here we can observe protein antibody DNA complex. That is the immunoprecipitation concept. Protein of interest interacted with an antibody, and we can take out this DNA sequence, amplify the DNA, and after sequencing, we can trace out the DNA protein interaction. This completes the chip assay. So this beautiful chip assay complete procedure is designed by my student Tejaswini Ritti from Shimoga. She has used 3 different protons, A, B and C. And when antibodies are added, antibody reacted with B protein, you can observe red color antibody and red color B protein. Once immunoprecipitation is achieved, we can amplify that sequence by PCR. So basically, chip assay deals with DNA and protein interaction.